So light energy strikes the photosystem. The photosystem contains chlorophyll. The energy from light, from the photons, causes the electrons to go up to raise energy level. They raise energy level and they are released from the photosystem. The photosystem passes it to the first carrier protein. This is a carrier protein. It is going to pass on the electrons to another carrier protein. Because these are passing on electrons and allowing the electrons to be transported, we call this first protein and then the second protein and then the third protein that are passing on these electrons, we call it the electron transport chain. And they are carrier proteins. So we can see 3 carrier proteins making up part of the whole thing, the electron transport chain. So going back, the electrons arrive at this carrier protein. Now this one's a special one because what it's going to do is use the energy to change shape, and it's working as a pump. This is a pump protein. It's going to pump hydrogen through from outside the membrane in the stroma to inside in the thylakoid space. So let's just pump some through. Those are our green electrons. These red are representing H+. They are protons. So remember, a proton has come from hydrogen when hydrogen splits to give H+ and 1 electron. So these hydrogen ions or protons get pumped through the membrane, ending up inside the thylakoid space. We'll come back to what they do afterwards. So we're going to build up lots of protons inside those thylakoid spaces and less outside. The electrons are then passed on to another carrier protein in the electron transport chain before being passed to our second photosystem, which is confusingly called photosystem 1, and the first one was photosystem 2. At photosystem 1, they get excited because light strikes and they go up to an even higher energy level. They're now carrying a lot of energy and they are passed on by carrier proteins eventually to an enzyme. And this enzyme here has the job of adding electrons from the electron transport chain and adding hydrogens, which are floating around, the hydrogen ions, the protons, adding those two things to an electron carrier. So this rectangle is representing that. It is called NADP+. Now NADP+ can pick up 1 electron, and it can also pick up, let's just turn that sideways, 1 of those protons. And when it picks up those, it becomes reduced NADP, which is also known as NADPH. So there are 2 names for this. This, as it's picking up, so before it didn't have an electron and a proton, this is NADP+. It's got a positive charge because to get like that, it would have given away the electron and the hydrogen. Now when we add electrons and protons to it, it becomes, so let's add them on, it becomes NADPH. This is also known as reduced NADP. So why is it reduced? It's reduced because of oil rig. Oil rig was oxidation is loss, reduction is gain. Reduction is the gain of electrons or of hydrogens, or in this case protons. So when our NADP, which didn't have these on to start with, when it gains those, it now over here, once it's gained electrons and hydrogen, it's now reduced NADP or NADPH. So why do we have this electron and hydrogen taxi? Well, it's going to take these high energy electrons and hydrogen over to elsewhere in the stroma, and it's going to be used to fix CO2. So we'll see that in the next step, but we need reduced NADP to fix CO2. It's going to provide the electrons and the hydrogen to take CO2 and to make it into a carbohydrate, to fix it in the form of a carbohydrate. Okay, so back to our chain. Let's just recap what we saw. We saw electrons raising up to a higher level, being passed along an electron transport chain. We saw that there were pumps, and these pumps could take hydrogen ions, protons, which are outside in the stroma, and pump them through into the thylakoid space to build up lots of these here. We also saw that then the electrons could be passed on, and due to light being absorbed in photosystem 1, they could be photoactivated, raised to a higher energy level before being passed on to further carrier proteins in the electron transport chain. At the end, we had what we could think of as being an electron and hydrogen ion taxi, which is going to picks up an electron from the transport chain, and from floating around, because this is an enzyme, so it's hitting the active site, picking up a proton, the taxi now can go off and transfer those to fix CO2. Now at the end, I've separated this with a dashed line because this is completely separate. At the end, and it looks on the picture as though it's together, this is completely separate, and we're going to deal with what that is now. Okay, so one product, we made our first product, which is NADPH. We've made NADPH or reduced NADP, and that is going to be used to fix CO2 in the next steps of the reaction. The other thing that we want to look at is, why did we pump these hydrogens? Why did we pump them there? Well, they were pumped there using the energy because now they will want to move, just like osmosis down a concentration gradient, they will want to move out to where the concentration is low. Here we have high proton concentration. Outside we have a low proton concentration. Now, because we've got a high to low, we know that we have got a gradient. We have high H plus if this is concentration of H plus. High H plus inside the thylakoids, inside the thylakoid space, and outside we have in the stroma a low concentration. So we could draw this as a graph. We could say that there is a concentration gradient. Now what hydrogen ions will do is move down the concentration gradient. They want to move down the concentration gradient. So they want to move from the inside of the thylakoid space to the outside in the stroma. Okay, so let's look at what happens there. So inside the membrane is this, and it's called ATP synthase. It is like a little molecular motor, like a little water wheel. If water turns round a water wheel, then with ATP synthase, as the hydrogen ions flow through, they cause it to turn round. And this is an unusual phenomenon because we can't really see anything like this happening in our normal everyday lives. But this is a chemical, or we could think of it being a chemical because it's H+, a chemical moving down a concentration gradient, which is causing a little molecular motor to turn. And this turning is used to add a phosphate onto ADP. There is phosphates floating around because ATP is always being used and broken down into ADP and phosphate. So this is floating around and ADP is floating around. And the turning energy is used as the energy to add them together and make ATP. So this is kind of strange, but by the flow of hydrogen ions, it turns this molecular motor round, and the turning is enough energy to put these two together and create ATP. And this is our high energy molecule. So what have we seen as the two products? Well, we've seen through passing electrons down the chain, we can pump hydrogen to build up lots of hydrogen which flow out through our molecular motor, turning it and creating ATP. That's one of our products. That's using the energy from light, which was transferred to the electrons, used to pump, build up a gradient, which flows out. Now, because this is the flow of a chemical from a high concentration to a low concentration, we call this whole process flowing from high to low chemiosmosis, because it's a chemical osmosis using the flow of protons which were pumped using the energy from the electrons. Now, at the end, we also had another product. Other than ATP, we also produced, by passing the electrons on, we produced our reduced NADP or NADPH. And we did that by adding a proton and an electron to NADP+. And that was going to go off and help to fix CO2. So now we've got both energy for the next reactions, and we've got electrons and hydrogen, which is going to help fix CO2.